Here the osmotic pressure of the solution, here it involves the term semi-permeable membrane. See the definition, a membrane which allows only solvent molecules to pass through it and not the solute particles or solute molecules. It is called as a semi-permeable membrane. Now this semi-permeable membrane may be natural or artificial. Number of examples are there. So it is a film such as cellophane, which has a pores that allows the solvent molecules to pass through it. But they are small enough not to allow the passage of larger solute particles through it. The examples, cellulose nitrate, animal bladder like pig's bladder, parchment paper, or artificially prepared semi-permeable membrane, copper, ferrocyanide. These are the examples of semi-permeable membrane. Now, what is osmosis? Osmosis, the flow of solvent molecules from the solvent to the solution, or from less concentrated to more concentrated solution through the semi-permeable membrane is called as osmosis. Now in your daily life, you can observe during the summer days, the raw mango, when it is placed in a concentrated salt solution, here it loses the water and shrivel into the pickle. This occurs due to the osmosis phenomenon. That is a common observation. Water moves from soil into the plant roots and subsequently into the upper parts of the plants. It is also due to the osmosis. Okay. Next, here you can well understood this osmosis phenomenon with the help of these diagrams, as well as after this diagram you will see the short video that can illustrate the phenomenon of osmosis. Now see here the definition of osmosis, that is here, what is the definition? That is the flow of solvent molecules either from solvent into the solution. So here you can observe solvent and solution, when they are separated by semi-permeable membrane, there is a flow of solvent molecules from solvent side to solution side. Or another alternative definition, if two different solutions with different concentrations are separated by this semi-permeable membrane, net flow of solvent molecule takes place from solution of lower concentration to solution of higher concentration. Try to understand. Lower concentrated solution means amount of solute is less. On the contrary, the amount of solvent or solvent molecules will be large. In this second compartment, the solution of higher concentration means the solute amount is higher. On the contrary, the solvent molecules are less. And this is the flow of solvent molecules. And therefore, the flow of solvent molecules from less concentrated solution to more concentrated solution. Here also, the same flow of solvent molecules from lower concentration solution to higher concentration solution through the semi-permeable membrane. It is called as osmosis. Now, if you remember, the first qualitative property, that is the lowering of vapor, relative lowering of vapor pressure. Second, elevation in boiling point. Third one, depression in freezing point. Now, elevation in boiling point, depression in freezing point, it is related to the first qualitative property, that is vapor pressure lowering. Similar to that, here also, this flow of solvent molecule occurs and it is favorable as the vapor pressure of solvent is greater than that of solution. Followed? So net flow of solvent molecules into the solution or from dilute solution to more concentrated solution through the semi-permeable membrane it is called as osmosis. Okay, as a result of osmosis, what happens? The amount of liquid on the pure solvent side decreases. On the other hand, here it increases. So solution becomes dilute as compared to initial concentration due to the osmosis. Okay, let us see the small video which is related to the osmosis. Here, I start this video.
This membrane separates pure water on the left from a solution of sucrose and water on the right. The membrane is semi-permeable. Water can cross it, but sucrose is too large for the membrane's pores. Water molecules will move from a solution with a lower concentration of solutes to one with a higher concentration of solutes. A solution with fewer solutes has more water molecules that are free to move about and follow their concentration gradient across a membrane. Thus, there will be net movement of water across this membrane from left to right. This diffusion of water across a membrane is called osmosis. Osmosis is of great importance in the water balance of all cells.
Now the osmotic pressure, which is the colligative property. Is it clear what is semi-permeable membrane? What is osmosis? Now the osmotic pressure. Now osmosis phenomenon, it can be demonstrated with the help of this experimental setup. So you can observe this experimental setup. Here, this one is the thistle tube. Now this mouth of thistle tube, it is firmly fastened or it is fixed by the semi-permeable membrane, that is cellophane paper. Now in this thistle tube, a sugar solution, it is filled. Then it is inverted, this thistle tube, it is kept in an inverted position in another container that contains the pure solvent, that is water. Now when the sugar solution is separated by pure solvent water by this semi-permeable membrane, that is cellophane paper, here what happens? We should expect that there should be flow of solvent molecules from solvent side into the solution through this semi-permeable membrane. So as a result of this, the liquid level in this thistle tube increases. Now it rises and finally stops. Now this liquid column in this tube, it exerts or creates the hydrostatic pressure. Okay. Now as the osmosis continues, here the flow of solvent molecule continues into the sugar solution, the liquid column, this level rises. When this level stops, finally a time comes where the liquid level in this tube stops. At this stage, the hydrostatic pressure which is developed due to this liquid column in this tube, here it is sufficient to force the solvent molecules back into the water. See again, here the liquid column in the tube, it exerts the hydrostatic pressure. Now this hydrostatic pressure, what it does? It pushes the solvent molecules back into the water which is in the outer container. Now at this stage, the hydrostatic pressure developed it forces the solvent molecules back into the water through this semi-permeable membrane. Okay. Now, here the equilibrium is established when the rate of forward and reverse passages are equal. Now, in this case, which is the forward passage? Forward passage means when equilibrium is established, means when the rate of passage of solvent molecules from outside container through the semi-permeable membrane into the sugar solution, this is the forward passage. When this rate becomes equal to the rate of reverse passage, means the rate of passage of solvent molecules from solution side into the solvent side, because hydrostatic pressure, it pushes these solvent molecules back into the surrounding container, that is the pure solvent water, through this semi-permeable membrane. And when these opposite passages, rate of passages, when the rates become equal, there is an equilibrium established. Now the height of liquid column at that time, it remains constant or it remains same. This shows that hydrostatic pressure, it has stopped the osmosis. Try to understand. Osmosis means what? When solution and solvent, here they are separated by semi-permeable membrane. The solvent molecules, that is water molecule from outside container, here outside, it enters into the sugar solution. So as long as this process continues, the liquid level in the tube rises. But this liquid column, it creates the hydrostatic pressure. And what it does, the hydrostatic pressure, it tries to push the solvent molecules back into the water which is in the outer container through this semi-permeable membrane. And when these both rates, that is rates of forward and reverse passages, become equal, at that time osmosis stops, means here the level remains constant. And this hydrostatic pressure which stops the osmosis, it is known as a osmotic pressure. Now it is denoted by the symbol pi. Now hydrostatic pressure, it is given by the formula H rho z, where H is the height of liquid column which is in the thistle tube, and rho is the density of solution, g is the acceleration due to the gravity. Followed this experiment, the concept of osmotic pressure. So what is osmotic pressure? It is the pressure, hydrostatic pressure, which stops the process of or phenomenon of osmosis, that is known as a osmotic pressure. Now consider the terms isotonic, hypertonic and hypotonic solution. Now iso means same. When two or more solutions, they exert the same osmotic pressure, it means that they must have same concentrations, then they are called as a isotonic solutions. Okay. Hypertonic means hyper. You know this word hyper means higher. If two different solutions are there, having unequal osmotic pressure means having unequal concentration. One is less concentrated, another is more concentrated. The more concentrated solution, it will have higher osmotic pressure. It is said to be hypertonic solution. Okay. What is hypotonic solution? Hypo means less. If two solutions have unequal osmotic pressure, and out of these two solutions, the solution which is more diluted, it exhibits the lower osmotic pressure. It is said to be hypotonic solution. Now here you can observe osmosis in a plant cell. So this is the isotonic situation. That is, here the solution outside the cell, plant cell, and inside the cell, the concentration is same. Therefore, isotonic. Hypotonic means here the outside this plant cell, here you can observe the water enters into the plant cell through the cell wall. So it means that here as water enters from the outside into the cell, it means that the concentration outside the cell, it must be lower concentration, therefore hypotonic. And this one is the hypertonic situation, that is the water from the solvent from the plant cell, it goes out into the surrounding solution. Means the surrounding solution, it must be concentrated, that is hypertonic. Okay, followed this concept: isotonic, hypertonic and hypotonic solutions. Now, before discussing the next point, that is the osmotic pressure and concentration of solution, it is better to see the one video which is related to the, here it is related to this isotonic and hypertonic and hypotonic solutions. So let us see the video so that that concept will be well understood to you. Okay.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So this is about the isotonic, hypertonic and hypotonic solution. Now osmotic pressure, as it is a colligative property, it depends on the number of solute particles or amount or concentration of solute in the solution. Here we say what is the relation between osmotic pressure of solution and the concentration of solution. Now osmotic pressure follows the equation pi is equal to N2RT upon V. Now in your textbook, the direct equation is given. So here, this equation actually becomes from the two equations, that is combination of Van 't Boyle's law and Van 't Charles law. Now in 11th standard, you have studied the gas laws, Boyle's law, Charles law. Here, Van 't, name of the scientist who stated this law, that is Van 't Boyle's law. Now the Van 't Boyle's law, here the statement osmotic pressure, that is pi, inversely varies with V or directly proportional to the concentration of solution at constant temperature. Van 't Boyle's. What is Van 't Charles law? Osmotic pressure of the solution pi, it is directly proportional to the absolute temperature at constant concentration, that is Van 't Charles. And after combination of these two laws, here it becomes pi, here N2RT is equal to N2RT upon V, where R is the molar gas constant. What is N2? It is the number of moles of non-volatile solute. Okay. Now N2 upon V, This term is the number of moles of solute per liter of solution. So that will be the concentration in terms of molarity. That is the capital M, moles per liter. So we can substitute this term N2 by V by capital M as it represents the molar concentration, moles per liter. So pi becomes equal to MRT. Now, in three colligative properties that we discussed in the previous videos, that is vapor pressure lowering, elevation in boiling point, and depression in freezing point, the solute concentration, it is expressed in molality. But here, the solute concentration, it is expressed in molarity. This is because the osmotic pressure measurements, they are made at a specific constant temperature, and therefore it is not necessary to express the concentration in terms of temperature independent quantity, that is the molality. Follow? This is the relation between osmotic pressure and concentration of solution. Now, molar mass of solute from osmotic pressure measurement. In previous three colligative properties, you have learned how to determine the molar mass of non-volatile solute from the colligative properties, that is elevation in boiling point, depression in freezing point, lowering of vapor pressure. Similar to that, for osmotic pressure, just we say this equation pi equal to N2RT upon V. Now, what is N2? It is the number of moles of solute. So number of moles of solute, it can be calculated by this ratio, that is W2 upon M2. W2, mass of solute dissolved in V liter of solution. What is M2? It is molar mass of solute. Okay. So here pi becomes equal to W2 RT upon M2 V. Here there should be M2. Now M2 is equal to, that is molar mass of non-volatile solute. Simply rearrange W2 RT upon pi into V. So by using this equation, here we can solve the numericals which are based on the molar mass of non-volatile solute from the osmotic pressure property. Now see the reverse osmosis. Next point, reverse. You know the word reverse. Osmosis. Now what is osmosis? The flow of solvent molecules from the solvent to solution through the semi-permeable membrane, that is osmosis. Or if two different solutions with different concentrations are there, the flow of solvent molecule occurs from less concentrated to more concentrated solution through the semi-permeable membrane. That is osmosis. Now here reverse osmosis means, here what happens when the salt water or seawater, when it is separated by the fresh water. Here the reverse osmosis means the direction of the osmosis can be reversed when we apply the pressure on which side. This pressure, it is larger than osmotic pressure. This pressure is applied on the solution side. So by applying the pressure on the solution side, here the flow of solvent molecule occurs from solution to solvent side through the semi-permeable membrane. That is, the direction of osmosis is exactly reversed, and therefore the phenomenon is called as a reverse osmosis. Do you followed this? The pure solvent flows from solution into the pure solvent, that you can observe in this figure, through the semi-permeable membrane. Now when this happens, when the pressure greater than osmotic pressure is applied on the solution side. Okay, this phenomenon, it is known as a reverse osmosis. Now you are familiar with this RO system that you are using in your house. RO system, that is water purifier. So reverse osmosis, this phenomenon can be used for desalination. Now D means removal. So removal of salts from the seawater. Here that is the process of desalination. For that the reverse osmosis can be used. The fresh water and salty water, when they are separated by the semi-permeable membrane, and when the pressure greater than osmotic pressure of solution is applied on the solution side, here the pure water from the salty water, it passes through the semi-permeable membrane into the fresh pure water. The process is known as a desalination process. Now related to this reverse osmosis, here we can observe the video, that is the reverse osmosis. Okay. This is the Adelaide desalination.
The core technology used at the Adelaide desalination plant is called reverse osmosis. First, let's explain what osmosis is. Osmosis is a naturally occurring process in which a liquid, such as water, spontaneously passes through a membrane. The membrane or semi-permeable barrier allows some molecules, like water, through, but other molecules, like the majority of salts, are unable to easily pass through the membrane structure. The flow of liquid through such a membrane occurs naturally to try and even out the salt concentrations between the two solutions. That is, the liquid flows from the less concentrated solution, such as fresh water, to a more concentrated solution, such as seawater. When the direction of liquid flow is reversed, it's called reverse osmosis. By pressurizing the concentrated solution, in this case seawater, we are able to force water molecules in the reverse direction, from the salty seawater side through the membrane to the freshwater side.
Okay. So today we see the fourth qualitative property, that is the osmotic pressure. How the osmotic pressure is related with the concentration of solution, the molar mass of solute, and osmotic pressure. From this osmotic pressure, the molar mass of solute can be determined, and the concept, that is the reverse osmosis. Now in the